Can't make any kind of reaction with cancer. It's a nice title. I fear that if I'm not. I was
Oh, you have no.
I can't have no because I bought some.
I'm gonna leave.
Okay, then I am using spices. No problem.
Cheers.
It's just reminding me, but thank God you're doing it. I don't want to ask you.
All right. Ready to get? Yeah. All right. Can everybody hear me? This mics. Okay, very good. All right. On behalf of the natural science faculty, I would like to welcome you to the 43rd annual Natural Science Research Symposium. A reminder of the college's mission is to develop students through scholarship, participation, and service. And what today represents is the culmination of 2 years of these students working through a capstone event where they are contributing scholarly work. They are achieving this level of scholarship in their major field of science. Okay. And so they've been working long and hard on this. This has been building to this point. And so I thank all of you for being here to support them and to ask questions of them and to help them dive deeper into their understanding with us. Just wanted to give you, like, this is the 43rd year. We've been doing this for a really, really long time. And there's been a variety of faculty members that have contributed to this exact same experience for year after year after year. Here's a list of the natural science faculty that have helped and done this since we began in 1981. And I just, we have a set of 6 of us that are working with students today. But I want to take just a second to acknowledge one in particular. Dr. Jonathan Fry has been here since 1993. And this is his last Natural Research Symposium as a faculty member because he's retiring at the end of the year. And so I wanted to acknowledge him for all of his help and all of his service. I would hate to guess how many students that you have advised in the 33-ish years that you have been a faculty advisor, but if it's not 100 plus, it's close to that. And so I thank you for setting the stage for all of this and to continue keeping going. This is a list. You have a program. There is a paper program on you as you walked in. You are welcome to grab one if there is still one available. But if you don't, and if you are more tech savvy, the digital program, if you would like a copy of that to give you an overview, of when we are going to have different presentations it's going to be every 15-minute intervals. The order of them is listed in the digital program as well as your paper copy. But it will also have a list of the abstracts for the presentations for each one of the students to give an overview of their research, a written abstract for you to review as you would like. Okay. All right. So with that, I will be up here helping shift between different students and I will be the moderator. So with that, I'd like to introduce Kyle Doberer. He is going to start us off with his presentation. So. Thank you.
My name is Kyle Dober. So my research project here was genetically engineering Saccharomyces cerevisiae to produce a protein to support the Nitroplast. Okay, so let's explain what all that is. So the Nitroplast is it's an organism or an organelle here in this organism. What you're looking at, the large piece there is what's called Ranger-Schmidt bigelowi. This is a single cell algae organism that's out in the ocean. It kind of floats around. It does it takes like sunlight, makes it into sugars with chloroplasts there that you can see. It also has inside of it a algae. It's called Candidiasis actylocyanobacterium thalassae, aka the Nitroplast. So the Nitroplast is a nitrogen-fixing cyanobacteria that's also just floating out in the ocean, but it also exists. It was thought to be an endosymbiont inside the algae organism, but according to the Science article that was published in 2024, they've declared it an organelle. This is because the algae organism produces proteins and it imports those proteins into the Nitroplast, so that way the Nitroplast can perform its function. So at the end of that, research article in 2024, the scientists hypothesized that they could possibly put the Nitroplast and the supporting genes that help make proteins for it into other organisms, possibly plants. This isn't anything new. Plants do this naturally. Up above me is a picture of a soybean plant or legume, and it has an endosymbiotic relationship with a bacteria called rhizobia. What this does is it produces the nitrogen for the plant, and then the plant also helps support by getting energy to the bacteria organisms. So scientists are thinking with genetic engineering, you could possibly transplant a Nitroplast into the plant cells and do some genetic engineering so that way they can be supported. So my particular research, I'm going to use, try and take one of those proteins from the algae cell, and I'm going to put it into a homolog that's also eukaryotic, and it's going to be yeast. So basically what I want to do is I want to transform S. cerevisiae yeast to express a protein derived from B. bigelowi to show that the organism can produce a protein that can be imported into the Nitroplast. That way the Nitroplast can perform its functions. So how I did this, we did a yeast transformation. We got this protocol from Millspore Sigma and used a lot of their materials. What this does is we take a plasmid, and which is the little red circular dots. This is circular DNA that can be put in to other organisms. And what we did was I put that into yeast cells or yeast colonies, and then grew those colonies. A really important part of this growing process is in order to separate these colonies that have the new plasmid, I have to insert a gene into it, which is URA3. This lets the new yeast cells grow in uracil deficient media. This is the vector that I created, or the plasmid. So the important parts of this is this has my protein, which is that green bar on your right side. This is the DNA that I was able to, I had to reverse translate this from amino acid sequence into a genetic sequence. Then I was able to create this plasma and order it from a company, and then I was able to insert it. In this plasma, there's also on the left side of that gene, the URA3 gene. This is what allows my new yeast cells here to grow into in colonies in the media that does not have URA3. From there, there's a promoter here. That's the GAL1 gene, which is the tiny blue one up at the top above the green. This is a promoter where I can take lactose and express that gene, and then the yeast cells would then produce it. However, we ran into part, which I'll get into. So my testing for this, we have 4 groups here. I had a positive control. Actually, I had 2 positive controls. One positive control had uracil in the agar, and one without uracil in the agar. The uracil in the agar was present just to make sure that I didn't accidentally wipe out my whole entire colonies because I did something incorrect. The positive control without uracil was to confirm that I did the transformation correctly. So if growth happens in this plate, essentially what it shows is that I did get plasmids with the URA3 gene into the yeast, and it was able to grow. There's a native control, so this confirms that there wasn't something that went wrong during, and there wasn't either uracil that shouldn't have been there, or perhaps what we'll find out is the yeast cells possibly already had the URA3 gene active in it. And then there's my experimental plate, which had my custom vector with my protein that I wanted to express as well as the URA3 gene to allow it to grow. So these are my results. I had to run this transformation 3 times. As I've kind of talked about, the first time that I ran it, I already had growth in all 4 plates. The positive control plates is what we want. The native control plate, however, growth in that one was not good. Because that means that there is somehow the yeast is able to get uracil produce it, and that can't confirm whether or not I've done the transformation properly. We ran the second time, and I made sure during the purification method that we made sure there's no uracil in the plates. While I was doing that, I was ordering another strain of yeast to ensure that I had a yeast strain that did not have uracil in it. That second test, again, we ran into the same issue, growth in all 4 plates. So by my third test, I have the results up behind you. I did have growth in positive plates, which is what we want. Negative plate did not have growth, also what we wanted. However, my experimental plate also showed no growth. So what I think happened here is the first 2 transformations and the time it took to order the new yeast strain in was about 3 weeks, which is about a week and a half too long to be storing my custom plasma. And I was pretty disappointed, but that's kind of how science goes. Sometimes you don't get results that you want. So if I were to redo it again, I would have that plasma and I would make sure to make back up. This is seems really simple, but it's something that I want when I was trying to study up the experiment. So having maybe half that vial back in long-term storage would have been a good idea. I didn't do it because I was hoping to just get results from the first time. The second thing I would do to improve my research, this goes on further, because I didn't get growth on that plate. I wasn't able to do the second part of research where I would do affinity chromatography and purify my my proteins, so that way I could either do a mass spec or an SDS page, which is where you're able to identify exactly the protein that you make. So, I'm not the only one that's probably working on this. In fact, I know I'm not, because, looking up, now, the sites that have, or the research papers that have cited my original article in 2024, there's about 200 of them now. A lot of these places are looking, or a lot of these papers are looking at, different parts, what you can do, with this, Nitroplast and the other genetic research that you can do. Two things that you can possibly do, like I've said, plants can be genetically modified, to produce nitrogen compounds. This is good for, using that instead of, possibly putting fertilizer. So this has, long-term consequences that I'll talk about here. The other thing is, yeast organisms, like what I was doing, can be genetically modified to produce ammonia. This would be a lot, possibly a lot simpler, process to do instead of, making ammonia the classic way, which is using the Howard wash process, which is a very energy intensive process. This is kind of the point, the reason for doing this. Agriculture, has two things. They want to, produce lots of food. It's important, and they need to do this because population is, increasing, by quite a bit. But by doing so, we are, using lots of resources to do that, including to do that, we have to use fertilizer. Runoff from those, fertilizer causes things like eutrophication, which is where waterways, get algae blooms. We've seen this, here in Kansas. This causes, a death of different organisms, plants, and animals in those waterways. Eventually, it washes down. This is a picture of the dead zone that's around the, Gulf of Mexico at the, Delta, Mississippi Delta, showing why this is important. We want to, help protect the environment. One of the ways that we can do this is by, doing, by doing this research, checking out, seeing if nitroplastic can be transported. So that's pretty much it. Acknowledgements, the Kansas Academy of Sciences, helps provide a grant funding for me, for this research. I'm really thankful for them. Dustin, Wilders, he wrote a letter of recommendation to the Kansas Academy of Sciences, help me obtain, funding. I'm really thankful for that. My research advisors were, Dr. Carrie Rathbone and Dr. Angela Orlagadar, and they helped me with technical parts. And then, the vector builder team is, the team that I got my plasmid from, and they helped provide some technical systems, for me to do my research. Thank you.
Are there any questions for Kyle?
Do you feel like that plasmid insertion was actually successful in your first 2 rounds, or you just can't say?
I can't for sure say it, that it was or not, because I'm pretty sure the yeast cell, that I was using, the yeast cell that I was using had, the uracil 3. So, I do think it was successful, but, I can't say for certain whether or not it was. It would be really neat to possibly go, and I've looked into what it would take to get, the Nitroplast ones, and that's really neat research. That I think would be cool. Yeah, I think this would be really cool research to follow up.
What was like the most difficult part of this?
I would say there's a lot of parts that were challenging. Parts that I enjoyed, that were challenging, finding and doing the, gene data research, and learning how to interpret everything. I found that challenging but fun, and then the transformation process took a lot of time. I was in the lab for I think 12 to 14 hours one day. That was very late, but that was great. So,
All right, thank you. Leo.
How's it going? My name is Leo Egging. My senior project aimed to look at the effect different stages of prairie restoration has on invasive plant species. My main advisor was Dr. Dustin Wilgers, and my co-advisor was Dr. Duncan Fry. So here's a map of native grass prairies in the United States, covered a wide range of the U.S. From the Rocky Mountains all the way to the Smoky Mountains. But due to colonization, infrastructure, and agriculture, native prairie is down to less than 4%. This is very important because native prairies provide natural habitat to wildlife, animals, insects, different species like that. Soil stability, with their root systems, developed root systems, mitigates soil erosion, and then the soil will then store carbon to mitigate climate change. Native grass prairies, their developed root systems, they could be 5 to 15 feet deep, so it keeps the soil in good structure. And the process of restoration is the process of reestablishment of native grassland that was once lost or degraded. So due to agriculture, infrastructure, or just lost prairie that hasn't been maintained. So here's a little diagram to show the different root systems. On the right are the native species, grass species. You see how long they are to help give the soil some structure. And then the non-native invasives only are about 1 to 3 feet deep. So they're very detrimental to the grass prairies. So an invasive plant species, what I'm talking about is an introduced plant from another country or ecosystem that brings harm to the environment through uncontrollable and rapid spread. And I aim to answer the question of how do invasive plants vary through prairie restoration stages. And my focus species were Cerevisiae lespedeza, field bindweed, and common ragweed, which is not an invasive plant species, but is a fast spreading noxious weed that I'll cover in a little bit. So getting down into the specific plants, Cerevisiae lespedeza, it was introduced for a good reason, to mitigate soil erosion, grazing for livestock, and provide cover for wildlife, but was soon realized that through the rapid spread and reproduction by animals, wind, and distribution, it's very detrimental to basically to native grass prairies. They prefer clay and loamy soils, which is very prevalent here in the Midwest. They're very tolerant to drought, acidity of the soil, and then shallow soils. And another reason why they're so detrimental is if not managed correctly, they will stay in the seed bank for up to 20 years. Here's a little image of Cerevisiae lespedeza. On the left is a younger plant. They grow through a single stem, and on the right, through time and reproduction, they'll very quickly flourish and grow in different stems. The next is field bindweed. They're common near high trafficked roadways and along agricultural fields. You'll find them in a lot of agricultural fields due to the constant tilling of the soil, because they don't grow in very deep soil. They're very resilient to any management approaches and survive in harsh weather, rainfall, drought, high heat, but they thrive most in moist soil where there's adequate rainfall. And then just like Cerevisiae lespedeza, instead of 20 years, they can stay in the seed bank for up to 30 to 50 years. A little diagram. So field bindweed, they grow a deep taproot. that figure A, you can see the mother plant growing that main taproot, and then they'll grow laterally through daughter plants and spread rapidly. Common ragweed. So they thrive in open, nutrient-rich soils, but best in areas with moderate rainfall. They tolerate drought and moderate shade, so really anywhere in the Midwest, they're very common. A reason why they're noxious weed, so they will reproduce within 2 to 5 months after emergence. So after their seeds germinate and they start growing, 2 to 5 months they will reproduce and spread. They'll spread by equipment, animals, as well as compost. Here's another figure of common ragweed. So my experiment was located at the Pear Farm, also home to the Kansas Sampler Foundation. They travel around the state to educate and support small rural communities. They have a number of agricultural fields that were restored to hidden grasses. And the lines that I've drawn show my transects that I walked. So I did the number 2 down here on the bottom left is the oldest prairie that I did back in 1983. And then field number 3 right there was 1993. And then field number 5 on the right was my 3rd field that I did. That was restored back in 2018. And then the biggest field, number 6, was just recently restored back in 2024. The reason for the different amount of transects is I wanted to keep the ratio of the size of the field kind of equal among the different prairies. So I made 50-meter transects for each transect, 2 to 4 transects per field. I use the assistance of an app on my iPhone called Picture This to help me identify different plants. The app uses data that has been inserted on their database to, if you take a picture of any plant, it will correctly identify whatever plant you were looking at. I reported different numbers of Stresa lespedeza and field bindweed for the invasive plants. And then, as I said earlier, although not invasive, it's a noxious weed, I identified common ragweed in the most recent disturbed field. So looking at my data table, field number 1 was restored 40 years ago. I did 3 transects in which I found 0 Stresa lespedeza, field bindweed, or common ragweed. Looking at field number 2, 30 years of restoration. I did 2 transects due to the size of the field. An average about 3.5 Stresa lespedeza plants per transect. Field number 3, back in 2018, I did 4 transects for size. Although not finding any Stresa lespedeza, I found plants of field bindweed, which averaged about 3.5 plants per transect. And then field number 4, the most recently disturbed, didn't find any invasive plant species, but identified the common ragweed as prevalent here. So looking at the older fields, the oldest field of 40 years back in 1983, they show strong developed root systems, which helps mitigate invasive growth. There's less competition for the native and invasive plants for space, nutrients, or sunlight. But then you get to the mid to younger restored prairies, where you will find a little bit of invasive plant species. Although there are structured root systems, there are still areas, open areas for them to invade in. And then the youngest field of 7 years or 1 year to 7 years, they just don't have enough time. They haven't had enough time for any plants, native or invasive, to germinate and then to allow root systems to develop. And I believe this is why common ragweed is prevalent in the early stages of disturbance, due to it being a fast spreading weed. with all the open space, no competition with any other species, they're able to grow. So anyone can do prairie restoration. It's not, you don't have to be a scientist to do it. But there are different factors into the process. You have to be able to identify early growth stages. If for a normal eye, any grass looks the same. But once you start looking at individual specific plants, the cerevisiae or field bindweed are going to stick out like a sore thumb. So different strategies for the management. For the early, most recent disturbed prairies, hand digging is going to be the top option, just because you're going to be able to identify the early stages of plants. And due to no other plants being around, it'll be easier to hand dig them instead of having to burn them, graze, or use herbicide. Once you have prairies that are 10 to 15 years old, there's going to be enough grass to grow and be able to do a prescribed burn. But all of these kind of go hand in hand together. You can't just do one without the other. Prescribed burns for grazing, grazers like cattle or bison or anything, they prefer grazing on recently burned areas for new growth coming in. Herbicide is good for small term practices, but after a while, if you just do herbicide, you're going to do more harm to the environment. So you kind of have to pick and choose on the different timelines and strategies of all these different managements, and understanding that from the data that we just saw, it takes at least 40 years for there to not be any invasives to be prevalent. So knowing it's going to be a long-term management, you have to be on it all the time, every year, for any invasive species to germinate and grow and spread. I'd like to thank the McPherson College Natural Science Department, all the way from the faculty to my advisors, and all the way down to my classmates for keeping everyone in high hopes for the past 2 years and today. And lastly, I'd like to thank the Penner Farm. Without them, this whole project wouldn't have been able to happen. They have an amazing farm, and you're able to go down there to see the different processes of prairie restoration, and educate people on the process.
I have a question. If you were to expand your study to include more transects or even more fields, not just from the Penner Farm, do you think that would affect your results, conclusions, what you've seen? How do you think that would change things?
So I believe doing more transects on different fields, knowing that there's only 2 invasive plants identified at this small farm, knowing that there are more invasive plants in the state of Kansas, I believe would be beneficial to. I know this was like a baseline project for me to do, but expanding on those different variables of different plants, where they grow, I think it would be beneficial to expand on different areas of the state as well. Some plants prefer different soils, different conditions. But yeah.
Why did you choose Penner Farm for research?
So Penner Farm, it was with the history of Penner Farm and McPherson College, giving anyone the opportunity to go down to study, gave me a good opportunity. And knowing that they did different stages of prairie restoration, knowing that there's from 40 years to as of recent, gave me a good site to do my project on.
Okay, bindweed questions. So bindweed has a deep root. So why is bindweed a problem, and where did it come from? And you can't hand dig it.
So field bindweed, it was introduced from Russia, um, with that deep taproot. In the summertime, just from experience, with my natural resource management, you can spray them in the early morning, but once the day gets going and they don't like the heat a whole lot. So they're very tolerant to any herbicide spraying. They'll close up their leaves, leaf follicles, flowers, and they're just very resilient to any kind of practice. So just spraying them one time is not going to do a whole lot. You kind of have to keep going at them over and over and over again.
Right? One more, real quick.
You mentioned using the picture. It's find it on that.
Yeah, it was very accurate. I've had experience to already do to practice natural resource management on cerevisiae and field bindweed. Just to make sure I used it just to make sure if I came across any other plant that I didn't recognize, I would have been able to identify it and add it also to my experiment.
Thank you, Leo. All right, next up, we have Amanda.
Hello, everyone. My name is Amanda Geering, and my topic is the effects of temperature and algae on duckweed growth rate. My advisor is Dustin Wilkers, and my co-advisor is Gary Batson. As we all know, climate change is very prevalent in today's world and today's society. It affects all living organisms on this planet. It affects every ecosystem that we know of, especially aquatic ecosystems. The producers in these aquatic ecosystems will be significantly affected, mainly because they use temperature to do photosynthesis. Now, photosynthesis is the process of turning light into energy for reproduction and growth. Now, plants have a thermal optimum of which they use the temperature in order to photosynthesize faster. And the best temperature for these plants is called the thermal optimum range or just the thermal optimum. It depends on the plant. Now, although all plants have these thermal optimum ranges, some of them do not grow effectively above that maximum temperature. As for the motivation, I wanted to do a freshwater ecosystem simply because they are readily available and very easy to access. The issue was choosing a species to work on, mainly because there are so many in these ecosystems. I eventually chose duckweed and algae. I needed the kind of next question to be able to form my hypothesis was how might these organisms operate in their ecosystems? Duckweed and algae live in the same ecosystem, thankfully for me, and they both compete for resources such as light and nutrients. Now, on the bottom, my left, there is this very bubbly type structure that is algae, and then everything to the left is duckweed. So you can see like these very small individual pieces. To get into some of the components of the organisms, lemon miner, also known as duckweed, is a freshwater water plant, and it lives in slow-moving water sources such as lakes, rivers, ponds, and streams. This plant reproduces asexually, and humans and animals are able to consume this plant. Duckweed absorbs nutrients to the underside of its frond and its root. We can see this in the bottom photo. This spaghetti-like structure are the duckweed's roots, and those are connected to the underside of the duckweed. The top picture is the top side of the duckweed, and each leaf-like structure is a frond. So this one has 4 fronds. Like I mentioned, duckweed does float on top of the water surface. It has an optimal temperature range from 20 degrees Celsius to 30 degrees Celsius. For those who don't know Celsius, the 30 degrees is 86 Fahrenheit. As for the next organism, it is Microspora, also known as algae. It is also a freshwater aquatic plant, and it reproduces asexually. Aquatic animals are mainly the organisms that consume algae, and it absorbs nutrients through every part of its being. So in the photo, algae is a very dark green structure. It floats on top and beneath the water surface. It has an optimal temperature range of 20 degrees Celsius to 30 degrees Celsius. As for my question, I wanted to know how temperature and algae affect the growth rate of duckweed. I thought that temperature would have a negative effect on duckweed growth rate once it got past 30 degrees Celsius. I also thought that algae would have a negative effect on duckweed growth rate simply because they are competing for the same resources. Now, as I was preparing everything, I went ahead and found a freshwater aquatic source, which was at the Hutchinson Zoo, where I was able to obtain my algae and duckweed. I then went back to the lab and created a water nutrient mixture, which consisted of 1 milliliter of G in a 5-gallon tank filled with water. Now, G is a substitute for nutrients that the duckweed algae would normally get in its pond. I filled the aquarium tanks up with water, and then I separated the duckweed and algae into different tastes, obviously, to have a pure sample. I then, of course, removed any obstructions such as moth larvae and debris, and then I would, of course, refresh the tanks with new water every week just to make sure everything stayed alive. I could grow my colony for my experiment. Now, again, there's another picture of duckweed up on the screen. That is what I considered, not really considered, but that was one duckweed. I took tweezers and grabbed 1 frond, and anything else that came up with it was counted as 1. So my fronds ranged from 1 frond to 8 fronds. It just depended on what came up whenever we grabbed it. Once everything was ready, I calibrated the environmental growth chamber to 30 degrees Celsius and a 60% humidity with the lights on 24/7. I then filled the sample cups up to 80 milliliters with the water nutrient mixture and placed 30 duckweed in all 40 sample cups. In 20 of those sample cups, I put algae, and then I randomly put 20 cups onto a tray, resulting in 2 trays for 40 samples. I did this randomly just to minimize any human bias. I put them into the environmental growth chamber, as we can see in this photo, and then exactly 7 days they were taken out. We then counted the number of living duckweed, and we repeated this process again, only switching the temperature to 40 degrees Celsius. Once I had all my data, I used Jamovi to run a 2-way ANOVA, and then graphed everything. This is the graph that I have, and for now we're going to focus on temperature. Temperature has a p-value of 0.001, which means there was an interaction. In order for there to be an interaction, I want my p-value to be less than 0.05. So this is really awesome. As we can see, the 2 lighter bars in 30 degrees Celsius A and 40 degrees Celsius B, the difference between those 2 shows that there is, like, temperature has an effect on duckweed. So as for our next one, algae, it had the same p-value of 0.001, which means that algae had an effect on duckweed growth rate. Again, this is also shown in the bar graph with the 2 darker bars, 30 degrees Celsius B and 40 degrees Celsius C. The difference in those 2 bars shows that algae affected duckweed growth rate. Since this is a 2-way ANOVA, it also checked for if temperature and algae had an interaction. This had a p-value of 0.208, telling us that they had no interaction. This is also shown in the graph. Now, if we look solely at 30 degrees Celsius C on bars A and B, we notice there is a difference between the 2 of them. Now, if we translate that difference into the 40 degrees Celsius area, we notice that the difference between the bars is similar. So the pattern basically tells us that there wasn't an interaction between algae and temperature. As for my A, B's, and C's, this is telling us what is statistically different and what is statistically similar. So for our 2 B's, the 30 degrees Celsius algae and the 40 degrees Celsius with no algae, this tells us that in those environments, duckweed growth rate was essentially the same. Okay, so just as a quick recap, algae had a negative effect on duckweed growth rate. Temperature had a negative effect on duckweed growth rate, and there's no interaction between temperature and algae. The increase in global and local temperatures, they're simply going to affect duckweed growth rate. Now, this could have cascading effects in the trophic system, with duckweed being unable to produce as much, and then all the predators of duckweed are unable to consume as much, which results in possibly some of them dying off. And predators of those carnivores will have less to eat as well in those moments that we die if they can't find another food source. Now, we can't say that duckweed and algae were competing because the algae in itself was not studied. So I highly recommend for future studies to examine algae and its temperature and the effects on algae, as well as adding different variables into this experiment until it simulates a true ecosystem. I'd like to give a big thanks to McPherson College Natural Science Department for funding this project, to my advisor, Dr. Wilders, to my little brother, Logan, for helping me count all the duckweed, to Dr. Rathbone, and of course, my family. Does anybody have any questions? No questions. Yes, Grant.
Why was it that counted the duckweed that came up with the individual frog?
Why was it that I counted the duckweed when it came up with the individual frond? Well, as it turns out, I guess I didn't do my research well enough, as most studies counted each individual frond, and we counted just whatever kind of came up. So that was a lack of thorough research on my part as to why we counted it that way. Any other questions? Yes, Thomas.
So does this still make it applicable to like the greater level of science?
Okay, so can I still apply it to science and the ecosystem, is what you're asking? In theory, yes, because we do know from individual studies that temperature affects duckweed growth rate, and that algae affects duckweed growth rate. So we could theoretically, it could have been higher or lower on my graph, but we'll be able to understand that it has, like, temperature has an effect no matter what. It's just how big was the effect or how small was it? Yes,
Could algae have any beneficial effects to aquatic plants?
that I cannot say, because I didn't study algae, I studied just
Just the
duckweed, right? So that would be a different experiment.
So
Yes.
your numbers all came from counts. Did you ever do anything related to like mass of the plant?
I did not, because I didn't think about it, to be honest, and spending so much time in the lab, just simply waiting for the environmental growth chamber was a pain as it already was. Any other questions? Yeah, okay, great.
We have a couple of minutes if you need to stretch your legs over your legs, so we stay on time for the people are coming in here in a minute or two, specifically for a presentation. We don't want to get too far ahead or too far behind, so we got just a couple of minutes before we should, should start. I want to get off schedule.
These chairs are so hard,
Yeah, I keep going.
and I'm like, right, simplified.
Those got. I know. Okay. That's yeah. When they came out. It is. And it was by themselves.
Yeah, I'm ready. Three times, I find. Because
That was the second.
Let's see.
I care.
Yeah.
We're starting in about 30 seconds. So. All right, we're gonna go ahead and get back on track again. Thank you for being a little bit patient with that. Our next presenter is Grant.
Hello, my name is Ryan Lucas, being presenting my research project for how drought conditions affect diversity and abundance in Kansas. Why study birds? Birds are a big indicator on the environmental health. When people notice changes in bird populations, it might reflect the health of the ecosystem around. Drought affects water and food resources such as insects, plants, other organisms. And I chose Kansas because it's in the middle of a major flyway, one of the 4 major flyways in the US, the Central Flyway. So I chose 3 habitats with 2 habitats, or with 2 locations in each habitat. For my wetlands, I chose Doberer National Wildlife Refuge, chose Cheyenne Bottoms. For my reservoirs, I chose Webster's Reservoir and Wilson's Reservoir. And for my grasslands, I chose Red Hills and Cimarron National Grassland. These 3, reason why I chose these habitats is they vary in water availability. The habitats differ in water availability and resources, which I expect differences in bird diversity since there's different abundances in water. I expect wetlands to have the highest diversity due to the high water availability. Reservoirs to be second, and grasslands to have the lowest. For my research question, does precipitation affect bird diversity and abundance? For my hypothesis, I hypothesized that more rainfall leads to higher diversity as well as abundance. And my strongest theory was wetlands and reservoirs have the most. For my data collection, I collected data using the Audubon Christmas bird count. It's a large-scale, nationwide, citizen-backed records, which individuals, not necessarily just scientists, but avid bird watchers can upload data when they've seen a bird or document when they've seen a bird. And it happens annually from December 14th to January 5th. For my precipitation data, I chose PRISM climate data. It collects data across the nation as well. State and federal sites use data from this place. It basically collects data from all kinds of weather stations around the nation. I looked at 30 years of data, 1995 to 2025, 6 locations across Kansas, and the thousands of bird observations. It was a lot of data, and multiple species tracked again. So I measured the total abundance of each year of the number of individual birds, species richness, which is the number of different species each year, and precipitation in November and inches. I grouped the birds into waterfowl and non-waterfowl. which I describe as waterfowl being dependent on aquatic habitats such as geese, ducks, herons, gulls, shorebirds, such. And for my migratory and non-migratory, I decided those would migratory would be species that travel long distances for breeding grounds and winter grounds. Non-migratory would be residential species. Waterfowl would be more terrestrial and less dependent on water availability. For my data analysis, I used Simpson Diversity Index, which measures species richness and evenness. For my statistical tests, I used one-way ANOVA school Wallace tests using Jamovi. And I used these tests to compare diversity across habitats, as well as differences between bird species or bird groups. So here is all four of the migratory, non-migratory, waterfowl, non-waterfowl, based on without the precipitation data. And you can see that migratory and non-waterfowl were most abundant in the reservoirs, grasslands, and wetlands. These graphs show the precipitation. The top graph, it shows bird diversity for waterfowl, non-waterfowl, and the red line indicates the mean precipitation in inches. It shows that there is not really correlation between precipitation and diversity between waterfowl, non-waterfowl, and then migratory, non-migratory precipitation didn't appear to have a difference or an effect on the diversity of these species. Some things that drives bird diversity. Habitat might be more important than the actual rainfall. Resource availability may influence where birds are, where they travel. These patterns, they might have occurred due to wetlands provide water and different food resources. And migratory birds, they depend on stopover points along the central flyway. And waterfowl, they're most mobile. They can travel between different reservoirs, wetlands, are able to go to different places, as water. So for my conclusion, habitat, each habitat is the strongest driver in abundance and diversity, not necessarily precipitation for these locations. So, and wetlands, they provided the most diverse, most abundance, and out of the three habitats. Some of the limitations with the Audubon Christmas bird count is, since it's citizen run and not scientifically run, anyone can put down birds, and depending on the amount of people, more people, they have more eyes and see more birds and count more birds. And another year, if there's less people, they won't see as many birds, and that has, luckily, the data there shows how many participants there were each year, how many party hours they had out in the field. And precipitation, it doesn't capture the full reason why diversity. There's the drought didn't show the temperature of these locations. It didn't show the soil moisture. It didn't show other species in these locations. That is one of the limitations. And my acknowledgements, I would like to thank Pearson College for allowing me to do this research project. But I would like to thank my advisor, Dr. Wilgers, and my co-advisor, Dr. Rathbone. I'd also like to thank the Audubon Society for providing all this data to me, as well as the Prison Group. I would like to thank Jamie Piasky for keeping me motivated through this research project. And then I'd like to thank Kate Hanback for emotional support through this research project. It was very stressful and she put up with me. So I would like to thank those people. So thank you. Any questions?
Did you try to look at just flat out numbers of birds? Or did you try to normalize your data by dividing by the number of hours of observation so you can compare each year to?
Yes, I just did not have that in the slides. But I did, that's another limitation that I have in actually determining abundance. Oh, Jay,
So did you see any like dips in bird observations in 2020?
Yeah.
So
there was a large dip, the amount of people, participants out in the field, collecting research or collecting data on birds. Some years, some people, there's way more people. They spotted a couple million birds of one species in that time. 172 different species. So there was definitely dips in years. Probably Loopy's right.
Yeah.
I was doing a research project over that, but it was a little too complex to take years to do, and yeah, pretty cool birds. But you had a question.
You selected precipitation a month prior to
Yes.
the collection. If you were to go back and either add more like water related data to your analysis to see if it was just you selected the wrong precipitation variable that isn't reflected, what would you choose?
I would probably choose humidity for maybe a month, a couple months before the count. If you count, use the data from December, it wouldn't affect the birds as much as London fans. So. Oh, Ty. All right, Ty.
I know these precipitation values. So did you ever, for the reservoirs, did you ever put the water level relative to the?
I was going to, but I couldn't. There was data on the water levels, but I tried to get just one variable.
Sure.
Instead of, because for grasslands, you can't, there's not really a
Sure,
water
yeah.
level for grasslands as well as wetlands. It's less. Can't really gauge water level on wetlands.
But just looking at it, did you see any patterns or anything that significant, at least without analyzing?
Reservoirs changed, and it was evaporated and ran. Tile.
Slide that said your hypothesis is not supported.
Yeah.
How is it not?
Because the precipitation over the years, it didn't affect the diversity or the abundance.
Okay.
Yeah.
That's the wetlands. It's not.
Just, yeah.
I'm constantly.
All right, next we have Zach. Oh,
All right, so yeah, I'm Zach Hawley, and my research was about the diversity of wildlife species which are affected with rabies in relation to urban and rural environments. My advisor was Dr. Wilders, my co-advisor was Dr. Manesel. Just a little bit of an introduction. I was born and raised on a small farm, in Southwest Kansas. I've grown up hunting and loving the outdoors my whole life, and so I wanted to combine that into research as much as possible, the outdoors as well as country lifestyle. So hence the rural and urban parts of this. Because of this, I came up with two hypotheses. My first one is the fact that there will be more tests in urban, suburban environments compared to rural ones. This is just because I believe in those more populated counties, there'll be more people to submit the specimens that he tested. Then my second one was the fact that there'll be higher positive species diversification in rural environments, just because if you think of rural counties, there's farmland, there's trees, and there's just more habitats for those animals. So what exactly is rabies? Rabies is a viral or virus that attacks the central nervous system, and it only affects mammals, interestingly, because it operates at a core body temperature of 98.6 degrees Fahrenheit. So this also means that it can be, humans can contract it. However, that also means, interestingly, possums cannot, even though they're considered a mammal, because their core body temperature is 94 to 97 degrees. So how do you get rabies? Mostly through bites and saliva, and also through wounds. It's a big factor is drool, is how it's spread. It's through direct contact. So that also means animals. When we get to the symptoms, main ones are aggression is a main one. That's because they have a lack of fear, and then also drooling, because like I said, it's spread through drool, and then rabies makes it to where the animals have a hard time swallowing, just because then just think if your mouth is dry and you're drooling and stuff, if you drink water, it disappears. But if it's spread through saliva, if the drool disappears, they can't spread it. So that's just the way it works. The other one is light sensitivity, and their day and night cycles are messed up. So say an animal is nocturnal, it'll be more likely to be out during the day and vice versa. As far as the symptoms, they appear typically 3 to 8 weeks afterwards. But after those symptoms appear, then there's about 7 days until the animal will pass away. So once symptoms show, it's a deadly disease. However, humans, there is a post-exposure prophylaxis, also known as PEP, that humans can get, but they have to get it within 72 hours or 3 days, and that will decrease your chances of getting rabies. Specifically in Kansas, the first case for humans was in 1921, and the last known case was in 1968. So what exactly did I do? First off, Kansas has 105 counties, and I split those into the rural, urban, and suburban counties, and that was based off the population. So here, none of the highlighted counties, or none of the highlighted counties are rural communities, and those are counties that have less than 100,000 people in them. The suburbs are the yellow ones, and there's only 3 of them. And so those are the ones that are ranging from 100,000 to 200,000. And then the urban are the orange ones, and those are the counties that have more than 200,000 people. So you have a wide range of
100 counties to 2 counties. So that will play a factor in. My specific areas of interest included the categories, which is the rural, urban, suburban, the number of tests, percent positive, number of species tested, number of species positive, and the Simpsons index as well. In all my tests, I ran through Jacobi, which specifically included the Kruskal-Wallis and the Dual-Steele-Critchlow-Klinger tests. So here's just a real quick breakdown of my data. Overall, I went through 11,515 tests, and this is over a 10-year period from 2013 to 2023. And of those 1,515 tests, 506 of them were positive, and 10,763 of them were negative. And then specifically when it comes to the species, there were 12 positive species and 43 negative species. And just interestingly enough, there was a negative test for a chicken in Russell County. And keep in mind, I said only mammals can contract rabies. So here's my first set of results. And this is the Kruskal-Wallis table. As Amanda mentioned, in order for it to be significantly different, it has to have a p-value of less than or 0.05 or less. So my specific variables that were significant included the average population and number of tests, percent positive, and the number of species tested. I specifically also want to draw your attention to the percent positive. It's the blue at 0.023, which is significantly different. However, when I went ahead and ran the Dual-Steele-Critchlow-Klinger, the percent positive here has a p-value of 0.069, 0.208, and 0.833. None of those are significantly different. So that was just an interesting difference, and I don't know what caused that. Because the Dual-Steele-Critchlow-Klinger tests, it breaks down your categories and it can compare any two at the same time. So when it came to urban and suburban environments or counties, there was no differences, and all of the differences came between the rural and urban and suburban. Then the next test that I ran, I have 5 of these tests. It's the spearmints, which just compares how 2 variables move with each other. I compared all of mine to the county populations. So as county populations increase, did the other variables increase or did they decrease? So in this specific instance, as populations increased, the number of tests also increased, which goes with my first hypothesis. Similarly, I again used county populations, this time with the percent positive, which this one actually decreased. So as populations increased, the percent positive went down. So while the bigger counties were submitting more tests, more of them were coming back negative. This also shows eventually the trend line would go down into the negatives, and while that is not possible, it will just level out at 0. Population, as the populations increase, the number of species also increased. So this is actually going against my second hypothesis. And similarly, the number of species positive also increased with populations. And then Simpson's index, this one shows the diversity between species. And actually, in this one, as the Simpson's index value gets smaller, the diversity increases. So in a way, this graph is a little backwards because it's showing as populations increase, the Simpsons index is also increasing, just because of the formula that I used. Because there are two different formulas that you can use for Simpsons index. And then through this research, I found skunks were by far the most positive. Skunks had a total of 622 tests, and of those 622, 320 of them came back positive. So that comes out to 51.45%. And if you compare that to the next highest one, which happens to be llamas, at 25%. And that's just because there's one positive llama test out of the 4 tests. So, and I noted Sedgwick County had the highest percent there. And I got that by taking, Sedgwick County had 26 positive tests over the 10 years. So I took that 26 divided by the 320 positive skunk tests. And here, throughout all the skunks, that relative percentage, when I took the county positive test divided by total positive test, was significantly different. So this brings me back to my hypotheses. The first one was the fact that there will be more tests in suburban and urban environments. This was supported. Just like I said, as communities increase, or as populations increase, the number of tests increased as well. But then it's also goes back to, even though there's more tests, less of them were positive. And then my second hypothesis was there will be a higher positive species diversity in rural environments. This was not supported. Like I said, the Simpsons index graph is actually inverted, so you needed a smaller number, or a larger number, sorry. I would like to acknowledge Dr. Wilkers, Dr. Van Selt, as they were my advisors and helped me through this. The Kansas Department of Health and Environment, as they provided me with all of my data, rabies, and all the tests, was 11,000. And then also the Kansas Division of the Budget, as they're the ones through who I got the population data.
What were the equations?
What were the equations? So the one I deduced was you take the sum of the total number of organisms in a species multiplied by the total number of organisms in a species minus 1 over the total number of organisms of a species minus 1 as well.
What's that? Fourth? Which one's that?
That's the one I used.
Oh,
Yeah. And then
well, you said there was 2 equations.
yes, that's the one I used. So then the other one, just in front of that equation, you just put a 1 minus. So you take the same thing, just subtract it from 1. Yes.
Any speculation as to why skunks are highest?
Yeah, what's with the skunks?
Yeah. They social players. So I didn't really find anything. I don't know why skunks were so much higher, because it's that's the other thing. It's a big conception that bats and raccoons are always high, and they weren't at all. So I don't know why the skunks were.
Were dark animals considered? Can I help?
They could be, yes. It's because in order, yeah. Dead animals and roadkill is just whatever the public submits.
If you were to broaden out your population, like your categories of 100,000 to, what was it, 200,000, like, if you were to like lower that a little bit to include counties like maybe like Saline County or McPherson County, or like some that have a little bit larger population sizes, but not quite at that 100,000 limit, do you think that might have changed things a little bit in relation to your hypothesis, or how do you think that would affect it?
Yeah, I do think it would change it. Just because I said those were also getting compared to like Wichita. So it's a lot more, I think the environments, I guess, and the habitats would be the biggest difference there.
Anybody else? All right, thank you. Right, next up is Brysen.
All right, good afternoon. My name is Brysen Kerby, and I did my senior research project over the effects of creatine monohydrate on C-peptide activity in humans. My main research advisor was Dr. Gary Rathbone, and my co-advisor was Dr. Dustin Jordan. All right, so starting off, why does this research matter? In the US alone, roughly 38.5 million Americans are affected by diabetes. It's one way or another. That's roughly around 12% of the population in the US. It's a lot. My research focuses primarily on type 2 diabetes, which is insulin resistant or insensitivity. Essentially, when we eat food, we turn food into simple sugar in our bodies so that we can use it for energy. And then insulin comes in so that we can regulate the blood glucose level. So when we're insensitive to this, right, it causes a whole bunch of issues. A lot of the times you don't. So it's obviously a huge health concern, but beyond that, it's a huge economic burden within the US. In 2022, we spent roughly $413 billion on type 2 diabetes treatments and things along that line. So why C-peptide? Because we're talking about insulin, right? So if you look at this graph behind me, it starts off with the precursor called proinsulin, where it leaves then into equal amounts of insulin and C-peptide. So the release in equal number of amounts. In undergraduate research, it's kind of hard to like do glucose testing or insulin testing because it requires drawing blood, taking plasma. And so I had to find an indirect measurement, a less invasive one. So I did some research and I found out that we excrete C-peptide through our urine. And so if we could get a measurement through C-peptide concentration in our urine, it would be indirect measurement that correlates to this concentration. And then why creatine? Creatine typically is used for muscle performance athletes that are trying to better their performance, get bigger, get stronger. But recently, there's been some evidence proving possible metabolic effects, specifically talking